Hello, and welcome to our next session, PBPK Regulatory Applications with Dr. Cecilia Tan. Dr. Tan is the Senior Science Advisor at the Health Effects Division at the U.S. Environmental Protection Agency Pesticides Program. She's actively involved in several PBPK modeling-related committees. For example, she co-chairs the Joint Research Center to develop OECD guidance on characterizing and validating PBPK models using the in vitro and in silico methods. And she's initiated several projects in the Health and Environmental Sciences Institute PBPK committee, including harmonized model reporting templates. I'm excited to hear what Cecilia has to say today, so let's go ahead and take it away.
Thank you very much. I'm so excited to be here to share some of our experience with using PBPK in our regulatory risk assessment, more specifically pesticide risk assessment. So, okay, let me, how do I, okay, sorry. Okay, so before I start, the disclaimer. Oh, I don't know if you can still hear me.
Yep, we can hear you and we see your slide.
Because the system told me to wait, it's not responding. So sorry about that. Okay, so disclaimer. So everything I say today is my own opinion, and it does not necessarily reflect the views and policy of the U.S. Environmental Protection Agency. Okay, next. All right. I apologize again.
Don't worry, is it not letting you advance your screen?
Oh, yeah. Okay, so today what I'm going to talk about is some of the examples using case studies to show you some of the common PBPK applications to support human health risk assessment. And then I will also share with you some of the common challenges experienced by both the modelers and the reviewers, and then some of our recommendations to address these common challenges. So these two figures that I'm showing here are generated using data from a PBPK knowledge base published by Dr. Judy Madden's lab in Liverpool University.
Oh, I'm sorry, Cecilia. It doesn't look like your slide is advancing, and we do want to see what you're set there with. So
it's advancing on my screen, so maybe I'll let you do it.
Sure. Just 1 second here.
I've seen the same issue with the other speakers too. So.
Well, there's nothing like doing things live and having these small hiccups. So our audience members, while you're waiting for the slide to come up here, we do have a poll question in the poll questions tab, just asking about maybe some of the future conferences you guys are interested in. So if you go over to the polls tab, you can answer the question there while we get the screen share here going.
So do you want me to try it again?
Yeah, go ahead and try it again. In the meantime, we'll still work on it on this side.
Okay. Oh, okay. You can see it now, right?
Yes. There
awesome.
We go.
Okay.
It must have been Josh coming online. Good timing there.
It must be.
We did it.
Okay, so let's go back to the presentation. So these two figures that you're seeing now, they are generated using the data from a PBPK knowledge base. It is published in this journal article. and it is from Judy Madden's lab in Liverpool University. And the purpose of showing these two figures is just to show you the chemical space coverage of published PBPK models. So the figure on the left shows the number of chemicals that have published PBPK models. I'm saying published because I know that there are a lot of models that are not published. And then the number of chemicals that have published models are very similar to the total numbers of botanicals or total number of pesticides. And then from these different databases, and it's about 1 to 6% of the chemicals in these different types of chemicals that have published models, with the highest percentage in, of course, drugs and pesticides, and the lowest percentage in rich chemicals, mainly because there are so many rich chemicals. And then the figure on the right shows the number of chemicals with PBPK models in different types of chemicals. For example, there are 392 published models for drugs. Okay, and next I want to use the EPA's Integrated Risk Information System, IRIS program, as an example to see how often PBPK models are used to support regulatory risk assessment, even though there are more than 1,000 publications out there. And a brief background on the IRIS program. This program derives human reference values for health effects resulting from chronic or lifetime exposure to chemicals. There are two types of reference values. Reference concentration is an estimate of continuous inhalation exposure to the human population, including sensitive subgroups that is likely to be without appreciable risk during a lifetime. And then the other type is reference dose, which is very similar to reference concentration, except that it is an estimate of daily oral exposure rather than inhalation concentration. And then using Judy's PBPK knowledge base, which I just showed earlier, we found that 147 chemicals out of the 571 that have complete IRIS assessment has published models, which is about 26%. And out of the 22 of these 147 chemicals that have PBPK, 22 of them mention the word PBPK in the risk assessment report, and only 14 of them involve the use of PBPK models. For the 18 chemicals that are currently being evaluated by IRIS, 10 of them have published PBPK models. Okay, so EPA uses different tiers of PBPK models in research as well as regulatory risk assessment, depending on the acceptable level of uncertainty in the application. So on the right side, for a more refined PBPK model, its predictability is usually evaluated using in vivo data, and then it is usually from animals, so that we can use a PBPK model for animal to human extrapolation, route to route extrapolation, or quantify human variability for within human extrapolations, or predict different exposure scenarios to interpret biomarker data. So today I will cover all 3 tiers of applications, but let's start with the more refined PBPK models. The reason we are doing animal to human extrapolation for environmental chemicals is that traditionally our risk assessment is based on animal points of departure, such as no observed adverse effect level, NOAEL, or benchmark dose level, BMDL. And then using these animal point of departure, we added uncertainty factor to estimate reference values for human exposure. Usually, default uncertainty factors of 10x are used to account for uncertainties and variability in animal to human extrapolation and within human extrapolations. As you can see here, there's a UFA, which is animal to human, and UFH is human to within human extrapolation. And when scientific information is available to account for animal and human differences, these information can be used to derive extrapolation factors instead of using the default of 10x. PBPK model is one of those tools that have been recognized to be a valuable means that can use to replace or refine default uncertainty factors. So here are some general criteria we use when reviewing a more refined PBPK models. One of the compartments is either identified as the target tissue, contains the target tissue, or is a good surrogate for the target tissue. Physiological parameter values should be realistic. Most, if not all, of the models submitted to us are encouraged to be written in programming language, especially open source language, and I'll explain more later why we request that. The reason we want it to be in programming language is so that we can check the model code for correctness. And then also we check the parameter values against their sources. We also evaluate how well the model predicts compared to available in vivo data, again, mostly animal data. And if the fit is not perfect, how does this difference impact the conclusions from the model? So uncertainty analysis. And going back to that iris assessment, if you remember, out of the 571 chemicals that have complete assessment, only 14 of them used PBPK models. In the first example, PBPK model was used to estimate an inhalation point of departure from an oral point of departure when only oral studies were available, so route-to-route extrapolation. And PBPK models are often used to estimate human points of departure from animal points of departure based on the same internal dose metric, such as using average rate of metabolism in liver for carbon tetrachloride, or peak blood concentration of a metabolite for this other chemical. And a PBPK model was used to replace the default inter and intra-species extrapolation factors for dichloromethane. And also, interesting, there is one example where a model was used to extrapolate from a 1 hour exposure at the low AL to longer exposure durations, so across different exposure scenarios. And then finally, in the case of methylmercury, the PBPK model was used to confirm or complement other methods to understand the kinetics of methylmercury. Okay, so if you remember the first slide where I showed the number of chemicals that have published PBPK models, you'll remember that no more than 6% of the pesticides or drugs have refined PBPK models. However, the agency still use PBPK models for other applications. In this next tier, in the middle tier, sometimes we use available ADME data to parameterize a PBPK model for hypothesis testing. For example, to analyze dose proportionality for study design or for dose response interpretation. Okay. So to optimize animal study design for a better result interpretation, data collection and model simulation should be an iterative process. On the left here is a list of common toxicity studies required for pesticide registration. So there are acute, subchronic, chronic studies, in vitro ADME, and then endpoint-specific studies like developmental, reproductive toxicity. And a PBPK model can be used to organize available animal data or non-animal data at any stage of the safety testing program to gain insight on the chemicals' PK behaviors, or to identify data gaps, and to optimize the design of subsequent studies. A model can be used to test hypotheses. For example, we can simulate alternative dose metrics. For example, peak concentration of the parent or AUC of the metabolite to see which dose metric correlates better with the observed toxicity data. A model can also be used as a tool to analyze dose proportionality to understand whether the kinetics is linear or nonlinear, and I will show you an example next. So in this example, we analyze the AUC, area under the curve data for 2,4-D, to see if kinetics can be used to explain the different dose response observed in male and female rats. In this 2013 dietary study, they observed treatment-related kidney weight change at 54 milligram per kilogram per day in female adult rats, but they did not observe any effect in male rats up to 79 milligram per kilogram per day. Their hypothesis for this lower NOEL observed in female rats is that renal clearance may be saturated at lower dose in female rats compared to male rats. And the saturated clearance is what leads to kidney toxicity. So they analyzed the AUC data from a subchronic study, 28-day study, by comparing the fold changes in doses and fold changes in AUC, which is shown in these two tables. For example, in male rats, from the dose from 5 to 79 milligram per kilogram per day is about 15-fold increase, but the AUC difference is about 40-fold, which is more than doubled compared to that 15-fold. And from their analysis, they suggested that in male rats, renal metabolism is getting saturated near 79 milligram per kilogram per day in male rats, and then around 26 milligram per kilogram per day in females, so the highlighted doses. However, there are several issues with this type of analysis, this fold change comparison. It is very subjective. So we probably do agree that 41 is different from 15, but is 10 different from 12. And also, this analysis used only the average AUC data, and it didn't account for data variability. So what we're proposing to do is using PBPK to test whether the data are better explained by linear or nonlinear assumption. And the PBPK approach offers many benefits. So here's a table comparing the PBPK approach with the other approaches. So other approaches usually assume a linear relationship between the external and internal concentration at the lowest dose group. But this assumption is not always true, and also it relies on only one data point, which is the lowest dose group. Using PBPK models, any a priori assumptions made in the model can be based on your knowledge about the kinetics, and then also the parameter values can be informed either by animal or non-animal data. The other approaches subjectively distinguish some dose groups to be linear and other dose groups to be nonlinear. But a PBPK model can simulate this continuous curve between internal and external concentration. So it doesn't define when it's linear, when it's not. And other approaches often use, like I said, average values. The PBPK model can incorporate all the individual animal data to account for either study or animal variability. And then finally, the other approaches analyze one data set at a time. For example, they look at AUC only, but a PBPK model can simultaneously optimize against multiple data sets. For example, like the two tables I show you, they have two tables, one for male, one for female, but with the PBPK model, you can simultaneously optimize, for example, KM for both sexes. Okay, back to our 2,4-D case study. Here are our model assumptions based on available data and knowledge about this chemical. Literature data show that 2,4-D is not metabolized. So in our model, 2,4-D is only cleared by renal clearance, no other clearance in the model. The model assumed either first order or saturable renal clearance described using Michaelis-Menten kinetics to see which assumption fit the AUC data better. We first fit the first order clearance rate, that's the linear assumption, or Vmax Km, which is the nonlinear assumption, to plasma AUC data from the 29 and 95-day studies for female rats. So we start with female rats first. And then we kept the same Km estimated from female rats and fit either the linear first order clearance rate or the nonlinear Vmax using male data. And here I'm showing only the longer term data and simulations for male and female rats. Male rat on the left, female rat on the right. X-axis is dose in milligram per kilogram per day, and Y is 2,4-D plasma AUC. The symbols are data, blue lines are model simulations based on linear kinetics, and the red lines are model simulations based on nonlinear kinetics. You can clearly see that for both male and female rats, nonlinear curve fits the data better than linear curves. And then we also tested, do a statistical analysis and confirm that nonlinear fit better than the linear assumption. The simulation also showed that renal clearance saturates at lower dose in female compared to male rats. I also want to point out, even though you see 4 dose groups for the female rats, originally there were 5 dose groups. The data at the top dose group were not available because excessive toxicity was observed in that dose group, and they have to remove the top dose group. And our simulation shows that this top dose group, which they eliminated, probably have significantly higher 2,4-D in plasma due to saturation of renal clearance. And here in this figure, I'm showing female rat data and simulation from the subchronic and chronic studies, 29-day versus 95 days. And the simulations assuming nonlinear kinetics. Using one set of parameter values, the model simulations were able to capture the AUC differences for different exposure durations, subchronic and chronic. The 2-week and short-term and subchronic. As I mentioned earlier, this study originally had a higher dose group in the 95-day study, but that top dose group was removed because of the excessive toxicity. So if a PBPK model was used to analyze the 29-day data and then use the analysis, the Vmax and Km optimized to that data to predict the 90-day AUC, they may see that a higher dose, larger than 80 milligrams per kilogram, may be too high. So they probably wouldn't need that top dose for the 90-day study. And this capability, PBPK capability to simulate different data sets for different exposure duration is something that the other approaches cannot do. Okay, so now for the highest tier application, the agency often use models with simple structure and minimum numbers of parameters to convert in vitro bioactivity data to external concentration or doses for chemical screening and prioritization. And the reason we're doing that is for environmental chemicals, most of them are not measured in the environment. And we have very little information on their exposure and hazard potential. And here's an example that EPA and many other institutes have investigated to estimate so that they can estimate the health risk from thousands of environmental chemicals that currently do not have any data. So in this approach, they are developing better exposure models to predict exposure, which is demonstrated as black lines on this figure. And the blue and red lines, the blue and red lines are exposure predictions. And then the black line are human points of departure converted from in vitro bioactivity data using high throughput model. EPA has a tool called high throughput toxicokinetic HTTK model that can allow you to predict external concentration from the in vitro bioactivity concentration using only two parameters, two chemical specific parameters, which is plasma protein binding fraction and intrinsic clearance. And then they can compare the human point of departure with exposure to see, and then using the distance between the two to identify chemicals that require further testing. Okay, so I have given you examples of how the agency uses PBPK models for different applications. And then for the second half of the presentation, what I want to share with you are some of the obstacles or challenges experienced by modelers and reviewers. And let's go back to the IRIS example again. Only 14 out of 571 chemicals uses PBPK in the IRIS risk assessment. And then here are some of the reasons why PBPK was considered but not used. The first reason is that some reviewers said model predictions were not validated with human data. We'll discuss this point more later. And then other reasons include models are not available, or it doesn't have the right routes needed for risk assessment. And in one case, a model was available but not needed because available PK data are sufficient to reduce interspecies uncertainty factor. So in this case, it's not that they do not want to use the model, but the model is not needed. Okay, so besides the reason that we just learned from the IRIS examples, there are other barriers. Qualified reviewers who are familiar with modeling approach and its application in risk assessment are needed within and outside of the agencies. And we need a user-friendly software for reviewers who are not a programmer to test the model. The third challenge is related to a reviewer's comfort level with predictive models, especially for environmental chemicals when human data are usually not available to validate model predictions. And the other two common challenges for the modelers are that they have to prepare different reports for the same model because different agencies have different requirements. And also a refined model can take a lot of time and resources to build. And for the reviewers, it's often challenging to reproduce or repurposing an existing model to fit their specific needs. So given the challenges that we just talked about, the rest of the talk will cover some of the our recommendations. The first recommendations is to provide training. There are some training opportunities through workshops and courses like CE courses at conferences, or courses offered at academic institutes, or workshops, for example, offered by software company like Simulation Plus. However, these opportunities may not be enough for a modeler who need to shepherd a modeling project from beginning to end, from problem formulations to model development, model evaluation, to submission of the model. It is often not enough for a reviewer either, who need to understand all these things, PK concepts, modeling, applications in risk assessment, to review PBPK submission in a timely manner. When possible, customized training can be offered to the users, and training on risk assessment can be offered to the modelers. And it doesn't always have to be formal. Sometimes it can be dialogues between modelers and users about the model use. And finally, the community need to come together to think of more creative ways to provide training and facilitate sustainable growth in the field. So some people suggested maybe there is a need for a centralized repository of training materials. Okay, and then the next recommendation is related to model validation using human data. Human data are often not available because of ethical reasons for environmental chemicals. And it is important to help the reviewer understand that we build models to make predictions. If data are available, then there's no need for a predictive model. It is like just, for example, if it's raining outside, you don't need a weather forecast to tell you that you will need an umbrella. Also, it is important to help reviewers understand that PBPK models are simplified representations of biological systems. So there's no need to make a model more complicated than needed, because we often heard reviewers say, oh, this thing is missing, that thing is missing, but those things may not be needed. And currently, there's no step-by-step criteria for model evaluation because each model is unique. But there are several guidance on best practices, including WHO guidance published in 2010 and the US EPA guidance published in 2006. And there is a new OECD guidance that I heard mentioned by other speakers earlier. It's published in 2021. And the main difference between the OECD guidance and the other two is that the OECD guidance focuses more on the approaches and applications using non-animal data or non-in vivo data. And here's a comparison of the three guidance. The EPA and WHO guidance cover very similar topics, except that the WHO document has a chapter on training, communication, and expertise. Also, both of these guidance cover the use of PBPK models for extrapolation purposes, and the OECD guidance demonstrate different model application using in case studies. And one recommendation for the PBPK community is to consider updating the best practices guidance every few years to include lessons learned from publications. For example, we are currently doing a literature review to understand the common practices for estimating chemical-specific parameter values for human without using any in vivo human data. An updated guidance can include new methods for parameterizing a model or to evaluate models, or examples of new applications. For example, like I mentioned, EPA and the WHO focusing on inter and intra-species and route-to-route extrapolations. And the OECD guidance has several case studies on in vitro to in vivo extrapolations. And there are other applications that would benefit from having best practice guidance, such as those proportionality analysis that I presented earlier using the 2,4-D example. And also, I mentioned that one of the challenges for modelers is that they have to prepare different reports for the same modeling analysis. different agencies or even different offices within the same agency may have different requirements for model documentation. Some agencies do not have a reporting template, so the modelers decide what is important to report. And a standardized template can reduce modelers' burden to prepare different reports on the same analysis. It is not just for the modeler. The reviewers can also benefit because they know where to find specific information, and the information is also presented more consistently, making the review more efficient and then also more timely. And finally, a standardized template can be customized to meet specific needs of different agencies. So there are several existing templates. For example, the Food and Drug Administration, FDA, and the EMA, European Medicine Agency, they each has a guidance designed for pharmaceutical applications. The WHO and OECD guidance that I mentioned earlier are designed more towards models for environmental chemicals. It's just that WHO is mainly for data-rich chemicals and OECD is more toward data-poor chemicals, but both have reporting templates in the guidance. And then this 2020 publication is based mostly on the FDA guidance by expanding it to cover both drugs and environmental chemicals. This is how different templates compare. The WHO template requires less information. The OECD guidance, because it is designed for data-poor chemical, so the discussion in the OECD template focuses more on the uncertainty of the model. The FDA guidance does not have a separate section on model purpose or supporting materials, but both are included in other sections. Earlier, we talked about the need for user-friendly platform for non-programmers to test a model. So let's just get to this first point here. Some agencies, like my office, pesticide office at the EPA, requires all models to be coded in open source software. The reason for that is most of the documents, if there's no CBI information, they are publicly available. And then also our risk assessment documents are publicly available. So that's why PBPK models submitted to us also have to be available to the public. Ideally, a model review panel should include different kind of expertise, including biologists, toxicologists, risk assessors, modelers, programmers. And it's often really hard to recruit modelers and programmers. There are many out there. And then another issue related to computing software is that a modeler can choose the software that they like, they prefer, right? And or what they are familiar or what is available to them. However, it is really hard for reviewers to be familiar with all of the software program or even have access to them. And also some of the older models are coded in legacy software, and they will need to be recoded or they may no longer be accessible. So there are a lot of issues with computing software. And here are some recommendations related to computational implementation. So again, user-friendly software. I know that there are some groups that are developing GUI so that it's easier for non-programmer to use the software. And model equations should be provided so that non-programmers can at least examine the accuracy of the mathematical representation of the model, even though they are not able to read model code. And then there's some suggestion that some sort of data exchange format like XML coupled with formal ontologies may help model documentation and translation. And finally, at least for models that are submitted to my office, we are recommending using open source software. And that is also why our colleagues at the Office of in the Office of Research and Development, they are also developing these open source tools like the HTTK model. And we also mentioned that reproducing or repurposing a model is challenging because models developed for academic interests are not often directly usable for regulatory purpose, especially when documentation is often insufficient to reproduce a model. And it is also hard to repurpose a model because it may not have the compartments that you need, or the uncertainty is too large for your specific purpose. And the second bullet point, model template code, it is different from the model template reporting template that we just talked about. That's a template for submitting models, but here we're talking about standardized ways to write mass balance code. And then I think the last point is extremely important, that the modelers are encouraged to work closely with the users from the very beginning so that they can make sure that the model that they developed can meet the specific needs. And also it is a way to help the reviewers understand the model early on, so they don't feel too intimidated when you submit a humongous package to them in the end. And so here are some of the recommendations that I presented today. We need to think about how to meet the training needs for both the modelers and the users. When we expand model applications, we need to update the current guidance or best practices. And developing templates, there are many reporting templates out there, and then there are some groups that are working on templates for model code. So to continue thinking about standardizing ways to do things can harmonize model development as well as model review. And finally, very important, we are encouraging more dialogues between modelers and users. That's all, and thank you for your time today.